Good evening, Ishika. How are you doing? Great. In your school, still this genetics chapter is going on, right? Have they decided on which type they'll be going for next, which chapter? Today we'll be finishing up this chapter. Next, I'm thinking if they are not still deciding your school, we can continue with our cell cycle chapter because that will contain meiosis and all, and that is still the part of your course, and it will become a good revision, or you can say good conceptual understanding for you for the you can say longer run in biology. Because meiosis concepts and cell cycle concepts, G1 phase, G2 phases, S phases, okay, that will be of great importance. Yeah, we'll be also be finishing up. So you can text me on Sunday and all whatever is comfortable to you, like which chapter they are going on. Else we will continue with our cell cycle, like what I have planned for you, because that will lay a good foundation for this chapter. Okay, so today we'll be talking about some special topics. Number one is the chromosomal aberrations. Okay, chromosomal aberrations. Chromosomal aberration or chromosomal disorder means there are two types of mutation I have told you. One is just scope of point mutation. You can just change the nucleotides, the base sequences in the DNA. Another is chromosomal mutation where you play with the chromosome number, where the nature, the evolution plays with the chromosome number, means the chromosome number doesn't stay constant. When you have a constant chromosome number, which is specific for a given species, constant chromosome number, that is known as euploidy. Ploidy means number, U means true. So the true ploidy for humans is 46 chromosome at the diploid stage, right? But we can deviate from euploidy due to effects like, or due to a phenomenon like aneuploidy. Due to a specific phenomenon known as aneuploidy, one can deviate from the euploidic stage, being means that it will not be containing the 46 number of chromosomes, it may contain lesser or more number of chromosomes. So the number of chromosomes that you can possess may be 47, maybe 48, maybe 45, maybe 49, etc. So you are breaching rule, the rule of possessing only 46 chromosomes, not more, not less, but you are possessing either lesser or more number of chromosomes compared to the 46 number of chromosomes, which make you eligible for being euploidic. That is known as aneuploidy. An means not. This is true. So this is false ploidy. So your ploidy number is false now. Okay. In plants, what we have something known as polyploidy. So that happens mostly in plants. Polyploidy events where you have chromosomal set change, means haploid chromosome in humans is 23, so you can have 3n, you can have 4n, you can have 5n. The entire set duplicates and it becomes added on to the, you can say, next generation of cells. Now coming on to the discussion that why do we have aneuploidic events? Okay, what is the reason? So let's delve back into the meiosis. When we start meiosis, we, that's why I was telling you that I need to teach you meiosis completely, so that in order to decipher this chapter completely, you need to have a great hold on meiosis. So in meiosis, when you start meiosis one, in meiosis one you have prophase one. Under prophase one you have the preleptotene, the leptotene, then zygotene, then pachytene, early diplotene, late diplotene, dikinesis. So many stages are there. So in the zygote in stage, what we have is we have something known as synaptonemal complexes. Okay, so the centromere of one homolog will attach with the centromere of another homolog. Okay, you have RUVC pathway. Okay, then you have REC BCD pathway. Okay, in the prokaryotes, we have RAD51 pathway. In case of eukaryotes, many enzymes are there which mediate the synaptonymal complex effect. Also, the mini chromosome maintenance complexes. Okay, then what you will be having is you will be having crossing over. The crossing over that I had shown you in some previous classes, I'll draw it again for you. Suppose we have chromosome from the father side. Okay, this one. Suppose this contains A, B, A, B. This one is the homologous chromosome from the mother side. Same chromosome number. Suppose this is chromosome number 1 from father. This is 1 from mother. Now A, B, A, B. Now crossing over means you will have something known as chiasmata formation like this, and then you will be going for crossing over events, means exchange of chromosomal arms. So this can cross over with this one. Okay, this can cross over with this one. Okay, so this is known as crossing over. So after crossing over, what do you expect to see? You expect A with this B and this B with this A. So I can say that after crossing over, what I'll be having is I can have this side will be the same A, B. This side will be capital A, but this will be small b. Now this side will be small a, this side will be same, but this one is capital B, right? So you can see that recombination has happened, but the distal chromosomes, they haven't taken part. So even if recombination happens 100 times, means every time you can say cent%, 100 out of 100 it is happening. Cent% of the time, every time recombination does not happen. So it is a thing known as recombination frequency. It's a chance event. It may take place, it may take place in the heterochromatic region between the same genes. Recombination may not take place between the more likely genes. The thing is, if at all between these genes only 100% of the time recombination happens, still the number of recombinants that you will be getting is only 50%. You can say that this is a non-recombinant. This is also non-recombinant. So out of the four, two are always non-recombinant. Maximum is you are getting these two are recombinants. So these two, so the recombination frequency what you will be getting is maximum. You will be seeing the recombination frequency of 50%. Okay, that is known as recombination. So after this recombination happens, this synaptonymal complex must be dissolved, dissolution must happen, and then disjunction or separation of the chromosomes should take place. Okay, at chiasmata. Okay, now if this disjunction doesn't happen properly, then what will happen? These two are there. If disjunction is not happening, that is known as non-disjunction. Non-disjunction. If non-disjunction happens, what will happen? You may have a break here, and these three chromosomes could go into one cell. This one chromosome may go into one cell, so that specific chronological order number will have a trisomy, three chromosomes in one cell, and one will have monosomy, that is, you will be having only one chromosome there. Okay, so what you will be having is one cell could have only one, and one cell can have three. One cell can have three chromosomes. Okay, so that means total number will become how much? 46 plus 1, here 47, here it became 45. So the answer is, why did, you can say, disjunction, or why did aneuploidy occur is because of non-disjunction in the meiosis stage, where meiosis 1, here also prophase 1. Okay, at packitine this happened, so at diplotin disjunction could not take place. Okay, at diplotin the disjunction could not take place, because of that this happened. And if you are unaware of diplotin packitine, Okay, that packaging we had chromosomal crossing over. Diploid we will be having the dissolution of the diploid will be having dissolution of the synaptonymal complex. So this dissolution is also mediated by a myriad of protein complexes. Okay, and plethora of other factors, but that will only be taught in the meiosis chapter because that's not a part of this genetics chapter. So next week when we will be dealing with it, I'll explain it to you completely. How does this stuff really occur? What is the mechanistic feature regarding diploid? Some mathematical tools are also give you to find out how to find out recombination frequency. Okay, how to find out the gene mapping, the location of the gene. Okay, those things I'll teach you also. That becomes very easy, and it makes biology really, you can say, conceptual in a way that you don't have to mug up anything. Okay, in case of genetics or meiosis. So this is the reason behind aneuploidy. So did you get the reason behind aneuploidy, Ishika? Any doubts behind the reason? Okay, good. Okay, so now we'll be going on to the types of diseases that can happen. Okay, so a chromosomal disorder occurs when there is a change in the number or structure of the chromosomes. Okay, so we have something like karyotyping. Okay, we have something like karyotype. I think you might have heard of it. I'll show it to you here. So karyotype is nothing but an individual's collection of chromosomes. Individual collection of chromosomes. So it's also a laboratory technique, okay, which produces an image of an individual's chromosome. It's also a laboratory technique that produces an image of an individual's chromosomes. So the karyotype is a very essential tool. This used to look for abnormal number or even the structure of chromosome. You can get a proper, you can say, portrayal of what is present inside the genome or in the genome of a specific person. Okay, so a picture of the chromosome is taken through microscope. In the picture of that chromosome is cut up and rearranged by the chromosome size. The chromosomes are lined from, you can say, the largest to the smallest. Then a trained cytogenicist, okay, he can look for missing or extra piece of chromosome. So I can just show you the karyotype. This is how a karyotype looks. You can see this is how any karyotype will look like. Okay. Just look here. You can see Y chromosome is pretty small compared to the X1. Okay. So there are 22 chromosomes. Okay. There are 22 chromosomes or 22 pairs of chromosomes which are known as autosomes. The 23rd pair is the sex chromosome, also known as the allosome. Okay. So each chromosome has been assigned a number based on its size. Okay, the largest chromosome is chromosome number 1. Okay, and you can say chromosome number, other chromosomes like 18, 19 are one of the smallest chromosomes in humans. Very important to know. Okay. So this is what you should be knowing. And one more thing I'll tell you is we have something known as short arm and long arm of chromosome. Okay, metacentric, then you have some metacentric, right? So you have long and short arm of chromosome. So short arm is known as the petite arm. Petite means shorter. Long arm is known as the Q. Q is the longer and Q comes after P, so it is the longer arm. So that is how it is labeled. Okay, suppose the chromosome is like this. The short arm known as the petite Q or P. Okay, this is known as QR or Q. So this is how the chromosomes are arranged. And here it is written as sen for centromere. Okay, here it will be tel for telomere. Okay, telomere repeats, which you will be knowing when I will teaching you the replication in the eukaryotic system. Okay, so now when we come to, you can say, the more or less you can say the numerical aberrations. Okay, I'm not going into structural aberrations. Structure will also deal with. Okay, so numerical aberrations when we get into. Okay, so numerical aberrations means the number of chromosomes, they will vary. The number of chromosomes in the person will vary. Okay, so it is also of two types, you can say, if you can describe it. Numerical aberrations, it is of two types mostly. Number one is constitutive. Constitutive means present in all the cells. All the cells, okay, due to meiosis. Because when meiosis occurred, the sperm only had that chromosomal defect. So the zygote had chromosomal defect. So every cell has that chromosomal number defect. Another is mosaic. So mosaic is disjunction after birth. Okay, so it is only present in some cells. Some cells, and when that is group of cells have become, you can say, defective, they will undergo mitosis and it may increase, but only a group of cells will be there which will be having the chromosomal defect, not each and every cell of the human body. Okay, so disjunction, I had told you, homologous chromosomes. Also in meiosis 2, the two sister chromatids separate at centromere. That can also cause problem. So that will be discussed in detail in the chapter, okay, the meiosis chapter. So come to number one. First of all, we can talk about sex chromosome aneuploidy. Number one is the Turner syndrome. You write it like this: 45x, or we write it at 45x naught. Both means the second partner of the X, either Y or second X, is not present. Only one sex chromosome is present. So the total number of chromosomes are 45, not 46. Okay. So what will happen is this is present in a newborn girl, which is not really a girl now because one of the X chromosome is missing. Okay. So the newborn will include redundant neck, Okay, skin. It can also have peripheral lymphedema. Okay, it can have congenital heart disease. Okay, by birth, that is congenital heart diseases. Then adrenal abnormalities. Okay, epinephrine or epinephrine secretion, hypertension. Then autoimmune, thyroid disease, obesity. Okay, and then the female will be sterile. Okay, little bit male-like development, less breast development, less secondary character. You can say development. All that will occur. Okay. One more thing. Next disorder would be the Klinefelter syndrome. This one is recognized by 47 because you have double XY. Instead of XY, you have double XY. Okay. So it is the most common cause of hypogonadism. The gonads, they do not develop properly, hypogonadism in case of males. And male will become infertile or sterile. Male infertility. Okay. It accounts for almost 10% of the males. Okay. Then if we come to autosomal aneuploidy, what we have is we have Down syndrome. This can come in the entrance or some other, you can say, objective questions. It is written as 47. Okay, it is 47, but trisomy in plus 21. Okay, this is how you can write it, because in an autosome it happens the 21st chromosome pair. Okay, just like that, it will have 3 chromosomes instead of 2. Okay, 3 chromosomes instead of 2, like this. Homolog will be there. Like this you had seen 21st, you can add one more. Okay. So you will be having 3 pairs in the 21st. So Down syndrome, I'll show it to you, karyotype. Karyotype of Down syndrome. Turner syndrome, anyone you can see. Just look here. Look at the 21st chromosome pair. You can see 21st. Okay, 3 chromosomes you can see here. 21st, 3 chromosomes. Okay, that means if you are a good cytogeneticist, you can discern or detect. You can see here, 21st chromosome, 3 chromosomes are there. Okay, so you can easily discern or detect, recognize the way it happens. Okay. So always know this for a fact. Okay. Now, what will be the ramifications? The consequence of having trisomy on the 21st chromosome. So what is happening here? All the genes which are present on the 21st chromosomes, they'll be coded more, because you have one more chromosome there. So that chromosome will also be expressed. The genes will be expressed, right? That will lead to dysmorphic facial appearance. Then flattened facial profile, flattened nasal bridge, protruding tongue. Then mental retardation. Okay. One more thing I'll tell you. There was a great research paper yesterday I was reading. In that, it was seen that the Down syndrome patients, the patients suffering from Down syndrome, not only have those patients have these kinds of abnormalities, defects. Okay, but they also seem to have hyperactivation of the immune response. So, hyperactive immune response. Why do they have hyperactive immune response? Because 21st chromosome, I told you have three chromosomes. So 21st chromosome, okay, also contains a gene which codes for interferon receptor. So if this gene is coded more cells will possess interferon receptor, which will bind to interferon which is secreted by the virus infected cells like interferon gamma, and that will cause more and more hyperactivation of the immune response. So this is one theory regarding hyper expression of the interferon receptor, okay, IFNR gene present on the 21st chromosome. So this is your Down syndrome. Next is you can have Patau syndrome. So here no need to know the details, just I'll tell you what happens. 13th chromosome trisomy. Okay. 47 plus 13. Okay, Edwards syndrome. Okay. You'll be having 47 plus 18th chromosome trisomy, just like Down syndrome. You'll be having Patau syndrome, Edwards syndrome. Okay, Turner syndrome also I'll show it to you, trisomy. You can see here, there is no Y or no X, X naught, okay, X0. No companion for X chromosome, that is the Turner syndrome. Okay, look very closely here. That is the Turner's syndrome for you. Then you have, if I type Klinefelter, look here, 2X and 1Y. You can easily make out that this is a chromosomal defect, a chromosomal abnormality, right? You can see here that is known as the Klinefelter syndrome. Then one more I'll show you, one very interesting one. Here it is only, cry-do-chat syndrome. Look here. Will give you good interpretation. You can see here, chromosome number 5 if you check. Okay. Look here. So look at this chromosome. This chromosome number 5, is it equally length to this chromosome number 5? No, right? The lengths of the two chromosomes differ. So in cry-do-chat syndrome, what happens is, this is also known as 5p minus syndrome because P is the petite arm. Okay, so it's a chromosomal condition which arises when a piece of chromosome number 5, especially from the petite arm, goes missing. Okay, and why is it known as cry-do-chat is because infants with this condition often have a high-pitched cry. Okay, which sounds like that of a cat. Okay, so cry-do-cat syndrome. Okay, so this is known as like that. Okay. So this is also a very good defect. Here structural defect you can see, not complete addition or you can say subtraction of a chromosome. It is not being erased or added up. Here a part of the chromosome goes missing, you can see. So this makes it even more interesting, right? So this is how the chromosomal defects they line up. Now when we talk about you can say the structural aberrations. Okay, that one was an structural aberration. Okay, so tell me, did you understand all these chromosomal aberrations? Okay, Ishika, any problem in subtraction or addition of chromosomes in all these diseases that I told you? Any problem in any of the disease, Down syndrome, Turner syndrome, Klinefelter, Edwards, Patau? Just some examples I gave you to make it more interesting. Cry-Ducat syndrome. Oh, okay, good. So now let's come on to the structural elaborations. So when we come to structural aberrations, what does it mean? It means that structural chromosome rearrangements, those are the structural chromosomal rearrangements. Some of the structure will be having some or the other kind of defect, like I just showed you in the cry-to-cat syndrome, where a part of the chromosome went missing, right? So structural chromosomal rearrangements are what, and how do they happen? They are the changes in the physical structure of chromosomes. Okay, and that may result in the consequences could be, you can say, birth defect, then mental retardation, then you will be having increased risk for infertility, then loss of pregnancy. Anything can occur. Now, due to, why does it happen? Due to chromosomal breakage or unequal crossing over. Okay, crossing over may not be equal. A part of the gene which was cut open, that part may not be, or that same, you can say, region or the amount or the length of the chromosome, I should be saying, it may not be reciprocated in the crossing over event, and then one chromosome may become quite long, one chromosome may be very, very short. So that could happen. Okay, so due to unequal crossing over, you can say, unequal crossing over events could be the true way you can describe, because like this and this, you can say go for crossing over. So it may not go for crossing over like this, it may go for like this. Okay, so what you can have is, you can have this has a very long segment and this has a shorter segment. So this one may become longer and this one may become shorter. Okay, like this. So unequal crossing over events may result in these kinds of specific dysfunctions or disformities in the chromosomal type. Okay, so what happens is, it could result in deletions, it could result in ring chromosomes, it could result in duplication, it could result in translocation. Multiple things can happen. Then it could result in insertion, then inversion. All these things can happen, okay, due to the result of certain damages, unequal crossing over and all. So deletions means the abnormalities in which a proportion of chromatin from a single chromosome is lost. Okay. If you talk about deletion, we are just talking in common way. Okay, we are not going really in depth because that is not a part of your syllabus. Okay, so abnormalities. Otherwise, we have specific mechanism, unequal crossing over, also mechanism governing unequal crossing over. Those things also we have. But I'm not going to go that deep. In which a portion of chromatin from a single chromosome is lost. That is your deletion. Just like I showed you in the cry to cat syndrome. Okay, it may be lost, right? Then we have duplication, very, very specific event. Duplication. Duplications are what? They are unbalanced rearrangements that result in partial trisomy. Okay, they are unbalanced rearrangements that result in partial trisomy. Now, they are believed to result primarily from unequal crossing over, especially in regions of genome where repeat sequences are found. Okay, that is how duplication of the specific DNA region happens. So we have a lot of repeats. We have mini satellites, micro satellites. Okay, so those repeats are always there. Okay, so that is the repeated sequences are mostly present in the heterochromatin region. But again, heterochromatin, euchromatin means histone acetylated, heavily methylated, deacetylated, non-expressive regions. Those have not been taught to you till now. Okay, so hold on to this one. Okay, when the molecular biology is taught, then I'll teach you extensive detail in extensive detail what duplication really is. Okay, and how does it happen? How does it happen during the replication? You can have this loop formation, the skipping, or the, you can say, repetitive, you can say, addition of the genetic sequences. So those things will be taught properly at that point in time. Okay, so just hold on till that time. I'll explain duplication and all in detail there also. Then we have translocations. So in translocations, what happens? It involves break into different chromosomes, not in homologous. Break into different chromosomes. With an exchange of segments. Segments. Now, in humans, what we have is we have reciprocal translocation, non-reciprocal translocation. Also, we have Robertsonian translocation. Okay, so reciprocal means like this. Suppose you have, show it to you. There is something as Philadelphia chromosome. That is a very good example of reciprocal translocation. Okay. What happens is you have chromosome number 9 here, and here another chromosome, not its homologue. Okay, chromosome number 22, suppose. This one is chromosome number 22. Okay. Now, this contains a specific region, a specific gene known as ABL gene. This contains BCR gene. So what happens? Some part of here will be connected to here, and this will be connected to here. So what you will be having is you will be having this one. Chromosome number 9, ABL gene is lost. Okay. Now what you have is you have here BCR plus ABL combination. Now you have ABL. This is 22. This is known as Philadelphia chromosome because it was first found in person living in the Philadelphia, United States, right? So the thing is, the result of this is what? This is the reciprocal translocation, means from both the chromosomes translocation happened. Okay, that is a reciprocal translocation. So what was the result is this ABL codes for a specific tyrosine kinase enzyme responsible for controlling cell division. And this BCR is a very strong, you can say, promoter, branch chain cluster or branch chain region, you can say. So this BCR region, okay, so this BCR region or breakpoint cluster region, it is mainly known as. So this BCR region, what happens is, when the ABL gene is very close to it, it will cause hyperexpression of ABL gene, and that will lead to excessive cell division and cancer production or cancer occurrence. Okay, so it leads to CML, chronic myeloid leukemia, a type of blood cancer, chronic myeloid leukemia. So that can arise due to this kind of translocation process. So it is a reciprocal. Why? This portion got cut from here and got, you can say, ligated here, but some part of this also got excised from here and got ligated here, but this didn't cause much change because this was heterochromatin, non-genic region, but this was a genic region. Genes are only 1.2% in the human genome. Okay, 98% is the non-genic region, more than 98. Okay, sorry, 1.8% is the gene. 1.8% of the chromosome is the gene, whereas 98.2% is the non-genic region. Or heterochromatin, this is the euchromatin, 1.8%. They are the non-coding regions, but they have their own specific advantage and significance both. They are just not junk DNA. You might have heard of junk DNA. They are known as junk DNA, but they are really not junk DNA. Okay. And then we have non-reciprocal translocation. Non-reciprocal means this is one chromosome. This is another chromosome. Suppose this part went and got ligated here, nothing got excised and ligated here. Okay, so the result will be this becoming longer, okay, and this becoming even shorter. Okay, that is non-reciprocal because part of this one got ligated here. Okay, that is non-reciprocal translocation. And the third one is the Robertsonian. So in Robertsonian translocation, what happens is, so tell you that. So it really happens in the acrocentric chromosome pairs, where one is very, very long and one is very, very short arm. Okay, acrocentric. Okay. It occurs in the acrocentric pair. Okay, mostly the 5 pairs of chromosomes are acrocentric. You can say 13, 14, 15, 21 and 22. Okay. They are acrocentric chromosomes. Okay, these pairs. Now, it involves the 2 homologous paired, or you can say non-homologous also. Both it can involve. Okay, and what will happen is, after the crossing over, Okay, or after the translocation, suppose it happened between chromosome number 13 and 14. Okay, this is 14. After this, what will happen is the partitioning will take place and the reciprocal translocation will take place. What will happen is the large arm will become larger and larger because the large arm has more genes. Okay, so it will become larger and larger, and the short arm also joins with the smaller reciprocal product, which contains only non-essential genes because that is a very short arm, and then it will wean off, okay, from the entire cell, okay, after cell division. So after this, what will happen is this and this will join, so it will form a huge chromosome, okay, almost metacentric, and these two will join, okay, they will form like this, very short arm. This may wean off, you may get one entire chromosome less, okay, but mostly genes will be retained, okay, because most of the genes are present in the large arm or longer arm, the Q arm, and this is a very short arm, so you can say very short P arm, okay, because that is an acrocentric chromosome. That is your Robertsonian translocation, okay. Then we have inversion also. Inversion means flippage. Just flip the segment. Okay, suppose this is the segment. You just flip the orientation. Suppose this is A, T, G, C. What you did was you flipped it. Okay, now it became C, G, T, A. This is known as inversion. If it involves both the arms of the chromosomes, okay, it will be a pericentric inversion, means involving both the arms, means cut in both the arms of the chromosome. Okay, one cut, then invert, then ligate again. Okay, it involves the centromere. If the cut is only in one arm of the chromosome, okay, one cut only, not two cuts, one cut only, invert, ligate. That is paracentric inversion. Here centromere is not involved. So these are the types of, you can say, structural aberrations that you may be able to encounter in most of your genetics experiments, okay, or genetic scenarios when you talk about the patients, okay, their karyotyping. So these things you can also deduce from the karyotype. Okay. So this brings us to the end of the genetics chapter for you. Now tell me if you have had any kind of queries in this chapter, any kind of doubts, queries, clarifications needed in this chapter, any concept or anything that troubles you. I think you are able to comprehend everything properly. Okay. So I've given you some extra details also for more conceptual understanding. Okay. So, okay. That's why I've kept it very conceptual so that you understand it from the biological perspective and you can solve any question. Okay. Yeah. Okay, Shika. So if you still have anything when you revise, you can definitely text me on my number and we can clear those doubts later on. Okay. Okay then. Thanks a lot. Have a nice day. Stay safe. Let's meet in the next class. Okay, Shika. Bye.